We are Group 19. This presentation will be focused on the role of cyclin A in chromosome segregation fidelity. This presentation is composed of three parts: the background information, and then followed by two research questions. What role does cyclin A have in microtubule kinetochore stability, and how does cyclin A degradation regulate the transition into anaphase? Cyclin regulates and drives different events during different phases of the cell cycle upon different cyclin CDK complex activation. As mentioned before, we will focus on cyclin A. As we can see from the graph in the left-hand side, cyclin A or S-cyclin accumulate in S and G2 phase. And from the graph in the right-hand side, we see its degradation begins in prometaphase and finish in metaphase. During prometaphase, the nuclear envelopes break down and the chromosome attached to mitotic spindle through kinetochore. Numerous kinetochore-microtubule attachment errors could occur in prometaphase. And if those errors didn't get fixed properly, it can lead to cancer. And with this, we're interested in knowing what role does cyclin A have in microtubule kinetochore stability.
Kabeche and Compton researched this question, and as shown on the left, using fluorescence photoactivation, they demonstrated that kinetochore-microtubule attachments are less stable during prometaphase, as shown through shorter half-lives. Cyclin A levels are also higher during prometaphase. And in order to determine the effect of cyclin A on stability, researchers supplied either a non-degradable mutant form of cyclin A or a cyclin A knockout to cells. With cyclin A levels overexpressed, as shown in the middle of the graph on the right, the stability is low regardless of cell cycle phase. And with cyclin A levels knocked down, the stability is much higher as shown on the far right. So this result indicates that cyclin A mediates kinetochore-microtubule instability. So why is this so important? Well, cyclin A enables time for processes to fix errors in incomplete chromosome attachment. As shown in the graphs, when researchers overexpress cyclin A, the proportion of cells in anaphase with lagging chromosomes is much lower. Therefore, increased cyclin A promotes chromosome segregation fidelity. This function of cyclin A is very important because chromosome missegregation can lead to aneuploidy that is common in many human cancers.
As we mentioned before, cyclin A is important for proper kinetochore-microtubule stability, and it operates in this manner under the regulation of the Spindle Assembly checkpoint. The Spindle Assembly checkpoint is a cell cycle checkpoint that is used in order to ensure proper chromosome segregation fidelity as the cell enters anaphase. The Spindle Assembly checkpoint regulates the degradation of cyclin A via a protein complex called the mitotic checkpoint complex. The mitotic checkpoint complex inhibits the ubiquitination of cyclin A by the APCC ubiquitin ligase. When the mitotic checkpoint complex receives a signal that all kinetochores are attached to microtubules, it stops the inhibition of the APCC, which is then able to ubiquitinate cyclin A and the cell can move into anaphase. This is how cyclin A is regulated in a cell cycle dependent manner. A paper came out a little while ago that used photoactivation to measure the turnover rate of microtubules, and they found that the microtubules were highly dynamic until the final chromosome had lined up on the metaphase plate. In order for these stable microtubules to form, it is required that cyclin A is degraded in prometaphase. This degradation of cyclin A must happen therefore very rapidly, causing the stabilization event to occur and for the dynamics of the microtubules to therefore change so abruptly. However, a paper came out a little while after that contradicted this. In this paper, they claim that the stabilization of the microtubules happens slowly and progressively over time as the chromosomes come together and line up in the metaphase plate. This would cause the SAC to deactivate progressively, and therefore cyclin A to be degraded progressively. This means that the cyclin A degradation rate mirrors that of the microtubule stability rate. With these two studies contradicting each other, more research needs to be done in order to find out the true mechanism for how cyclin A degradation leads to stable microtubules.